Namaste everyone. Welcome to our next class in mechanical properties of fluids. As you see on your screens, we are having a very interesting objective today. That's to study about capillary action in liquids. And what I would like you to achieve at the end of the class is that you understand the reason behind capillarity in liquids. You will be able to derive an expression for how high the liquid will climb in a capillary tube. That's what we call as ascent formula. And if from now on you are going to look at certain situations or occurrences in your real life, you should be in a position to illustrate capillarity in such cases. So let's begin our lesson with the definition of capillarity. Now, what is meant by capillarity? When I'm going to insert a tube which has an extremely smaller diameter in comparison to the container in which water is kept, that's extremely small, a very narrow tube, then the liquid tends to either rise up or fall, against gravity. Then we call such a case as capillarity, as you see on your screens now. There are two examples that are shown on the screen. One is water to your left and the mercury on your right. Now you can see here that when a very narrow tube is going to be inserted, we have shown two tubes of varying diameters. If it is going to be inserted in the liquids, you can see that water is elevated, that water is pulled up the tube. Water rises up the tube. Whereas in the case of mercury, if you see, mercury is not elevated, rather it is depressed. It falls down. Water rises up while mercury falls down. Now, why does this happen? We all know that liquids exhibit both cohesion and adhesion. Now, in the case of water, we know that the adhesion of water with glass is more than the cohesion between themselves. So what happens is they get stuck to the glass surface while the cohesion is unable to pull them down. Whereas in the case of mercury, cohesion is greater than adhesion, which makes the liquid to pull itself down rather than to be pulled up by the glass tube. We will also look at the reason for capillary in a different way. For now, that's what is capillarity, the rise or fall of a liquid in a tube of a very small diameter is what we call as capillarity. And such tubes are what we call as capillary tubes. Yeah, the tubes with the capillary tubes with very narrow diameters, very small diameters is what we call as capillary tubes. Now this rise or fall happens because of surface tension. Also there is a balance between the surface tension that is acting on the free surface of the liquid and the forces that are acting on the liquid surface. Apart from surface tension, that is going to be the gravity and the force, the pressure, not the force, the pressure that is acting on the concave side. All these go to result in the capillarity in liquids. Please understand this rise. If there is going to be a capillary rise, it is going to be against gravity, because gravity tries to pull it down while it tries to be moving up the capillary tube. Let's look at it mathematically to understand why this happens and how far can a liquid rise in a capillary tube that is being provided. So we proceed to derive the ascent formula in this case. So let's move on. Suppose I'm going to consider a trough. Let me make it a little thinner, right? Suppose I'm going to consider a trough which is filled with water. Now we know that if the trough is going to be big enough, not too big enough, that you would have seen it while you were doing various experiments in surface tension, a slightly wider cup or a slightly wider plate, you are unable to see the concave meniscus. Actually, that's because the radius of curvature becomes so big that you actually fail to see the curvature, but the curvature is still there. Please understand, it behaves like a stretched membrane, and the free surface actually has a skin-like behavior, which there will be adhesion over here more, while there will be cohesion more over here, and hence there is going to be a slight curvature formed. But because of the width of your cup or your plate, we do not generally see the curvature. So let us consider one such trough, and let me consider water to be filled to such a height. What is going to happen when I'm going to insert, I'm going to give you a very thick tube here, but kindly understand this is only to understand the mechanism that is happening within the tube. And please get that very clearly, that the bore of the tube is extremely small, extremely small. Is that clear? Even a thicker straw cannot be considered as a capillary tube, because the diameter is considered to be bigger than that is to be meant for a capillary tube. Now, once I am going to introduce this tube inside the liquid column, outside the capillary tube, the liquid layer again forms a greater cohesion here, greater adhesion there, and cohesion here. Right? So you are going to have two layers formed over there. But what happens inside the capillary tube? Now listen, let us assume that here it is going to be air. Let us assume this to be water, and this is air-water interface. So the pressure above is atmospheric pressure. But we know that for any liquid surface, there is an excess pressure over the concave side, which is 2T by R. The pressure is excess on the concave side over the convex side by a factor 2T by R. Isn't that clear, children? We have already done this a couple of classes earlier to calculate the excess pressure over a curved surface. So we have an excess pressure 2T by R. Now, so that means the pressure on the convex side is less by a value 2T by R. So that means if I'm going to consider, let me use a different ink. If I'm going to consider a point here, if I'm going to consider a point here, and if I'm going to consider a point here, they all should be at atmospheric pressure. But if I'm going to consider a point slightly lower, if I'm going to consider a point here, it is lower by 2T by R. So that means if I'm going to consider a point here, which is expected to be at PA, because according to Pascal's law, we need to know that all points which are at the same height in a given liquid column should be at the same pressure. But because we are within the liquid here, there is a pressure 2T by R, which is less. And we are talking about liquids in equilibrium. So in order that the liquid surface stays in equilibrium, in order to balance for the reduction in pressure due to the concave and the convex surface of the meniscus, the liquid surface will move up, so that this point within the liquid surface is going to be at the same pressure as PA, because PI is going to be PA minus 2T by R. The pressure within the liquid is less by 2T by R over the pressure that is outside the liquid surface. So in order to balance this difference that is existing within the liquid surface, the liquid is going to move up. Now, how far will it move? How far will it move? It will move to such a height. It is going to move to such a height. Let's look at that figure once again. And let's look at a capillary tube. Water here, water here. Let's assume it rose to this level and stopped. What determines this height? Now let us assume a point over here, which is at a height h from the free surface of the liquid. And let us assume that the liquid has a density rho and the gravity at that point is g. By Pascal's law, what will be the pressure at that particular point? Let's assume this point is A. What will be the pressure at that point? The pressure will be H rho G. This also we have done in the classes where we discussed Pascal's law. So what happens? This height that it rises to will be increasing the pressure at that point to H rho G. But what did we see? We saw PI is equal to PA minus 2T by R. So how much did I want it to increase? I wanted it to increase by 2T by R, so that I wanted this, the pressure inside, to increase by 2T by R, so that the pressure at this point becomes equal to pressure at this point, which is PA. Isn't it, children? It has to become that equal to PA. So that means this H rho G, which was gained by the liquid by rising up the capillary tube, should be equal to 2T by R. So that gaining has to be equal to 2T by R. So the height gained by the liquid will be in such a way so as to balance the difference between the internal and the external pressure. That means my H rho G becomes equal to 2T by capital R. Now, what is this R in our expression of excess pressure? Please understand, this is the radius of the meniscus under consideration. Now, let us look at that meniscus and the radius consideration a little bit closer with a wider view so that we get to know. So let me consider such, let me expand it slightly so that you get to know it clearly. So let me assume that this is my meniscus. This is the meniscus, sorry. This is the meniscus of my liquid. I'm considering a concave meniscus, and this is the height to which it has climbed. Let us assume the water level is over here. So this is the height to which it has climbed up. And I have told you that we are considering a liquid of density rho placed in a position where the gravity is G. Now, let us assume that the center of this curvature is here. So if I am going to draw a line like this, this becomes capital R, which is the radius of curvature of the meniscus. Now, let me draw a tangent to the meniscus at the point of contact between the surface of the glass and the liquid. So what am I going to call it as? This tangent becomes the tangent at the point of contact. So this value theta, which is my angle of contact. We have done this in the previous class. If you have not watched it, kindly go and look at what is angle of contact. Now this becomes my angle of contact. Now, what will we know that for by geometry, if the liquid meniscus is going to be formed within the capillary tube, this distance is going to be the radius of the capillary tube. It cannot be other way. The center of the capillary tube and the center of the meniscus will be one and the same. So I'm going to leave something up to you. We know that this angle is going to be 90 degree because the radius is always perpendicular to the tangent at the point of contact. So if this is going to be 90, this value is going to be 90 minus theta because the other angle is theta. But we know that this is also 90 degree. Yes or no, children? So we have this particular angle, this angle at the other side also 90 degree because it is going to be the angle between the normal and the radius at the point of contact and the tangent. So what happens over here? Now, by vertically opposite angles, I can mark this also as theta. And just now I said the other angle is also 90. So obviously this also becomes 90 minus theta. And I'm going to clear off some of the angles for you to finally write the answer. So if these two are theta, this is 90 minus theta. I want to understand, I want you to understand that this angle becomes equal to theta by ASP of a triangle because I have dropped the radius of the capillary tube from the center to the capillary tube. That's going to be a perpendicular that I have dropped because that's the shortest distance that's going to measure the radius of the capillary tube. So I'm going to erase off unnecessary angles over here and I'm going to hold theta over there. And what is theta for me here is the angle of contact. Now, if I'm going to consider this, let me consider this point as P over here, this point as L over here. So I have a right angle, the triangle OPL, in which I'm going to try and apply some trigonometry and write cos theta is equal to small r by capital R. So where I get the radius of curvature of the meniscus as small r divided by cos theta. Now, why did we take so much of explanation to get my capital R is equal to R by cos theta? Because to measure capital R experimentally is going to be a little difficult, but very definitely I will know what is going to be the radius of curvature, the radius of the capillary tube because the manufacturer is going to provide me with that particular data. And for a given liquid, the angle of contact with the particular material is a constant given some standard conditions of temperature and the purity of the liquid. So what have we derived that now? We have reached capital R is equal to R cos theta. So what were we initially writing about the link between the height and the radius of curvature of the meniscus? H rho g is equal to 2t by r, where t is the surface tension offered by the liquid. Now, what did we find R to be equal to? We found R to be equal to small r divided by cos theta. And I'm going to proceed to substitute that over here. So I get an R and a cos theta over here. So what am I going to write about the height to which the liquid will climb? That will be equal to 2t by r rho g into cos theta. T, surface tension of the liquid, R, radius of the capillary tube, rho, the density of the liquid, and G is going to be the gravity at that place. So you can see that H is going to be majorly dependent on cos theta, provided you keep the other values a constant, where theta is the angle of contact. So what comes into picture now? We are going to discuss about this capillary rise, this ascent to with regard to the angle of contact. So let's move into that. If I'm going to take my first case, theta is acute. Now, when will theta be acute? Pause the video and tell yourself. Yes. Theta is acute when there is going to be adhesion between the liquid and the solid surface greater than the cohesion between the liquid molecules. That is a very good example, water. So when theta is acute, the liquid will wet the solid surface. Cos theta will be positive. Cos theta is going to be positive. If cos theta is going to be positive, H is also going to be positive, because H is equal to 2t by r rho g into cos theta. Everything else is positive. My surface tension, radius of the capillary tube, density, gravity. We are going to consider gravity to be positive always. So my H is positive. That means the liquid will have a capillary rise. It will be elevated when a capillary tube is going to be inserted inside the liquid surface. Now my case 2, if theta is going to be obtuse. What's going to happen if theta is going to be obtuse? Theta goes to the second quadrant, cos theta becomes negative. That means my H also becomes negative. When H becomes negative, that means the liquid is not going to rise, rather the liquid is going to fall. There is no elevation, rather there is depression. A good example is mercury, which has an obtuse angle of contact, and hence if you see capillary tubes being inserted in mercury columns, there will be capillary depression, there will be capillary drop and not capillary rise. So we have justified mathematically also that liquids will either rise or fall in a given capillary tube depending on their cohesive and adhesive nature in the solid in which it is in contact with. So that's about your ascent formula. The different cases where you can understand why a liquid either rises up or falls down, and why at all should this happen is because the liquid tries to be in equilibrium. Excuse me, the liquid tries to be in equilibrium and tries to balance between surface tension and the force of gravity in order to stay in equilibrium. Now, the next thing that we are going to see, and some interesting thing about which we are going to have our updating the DP as our next activity, will be capillarity in real life. By now, most of you would be actually, knowing what's capillary. So I'm presenting a few examples over here. A beautiful example over here, a beautiful capture also is capillary action in the burning of a candle. So you can see here there is little molten wax over here due to the heat. So this melted wax actually rises up the wick by capillary action, which acts as a fuel for the burning of the candle. Now, the same way is our lamp over here. So the oil moves up the cotton wick by capillary action, again providing fuel for the burning of the lamp. And this is one example which we did during our Science Day exhibition last year. This beautifully shows capillary action of water along the tissue paper. So you can see here colored water rising up the tissue paper and showing the capillary action against gravity. You can see here that water has risen up the tissue paper, which again shows capillary rise. Now, this is another experiment your biology teachers would have shown in order to show the conduction of water through xylem vascular bundles and phloem, where they show the sucking up of water by these conduction tubes. So here you can see they are all three same leaves, same plant stems that have been placed in three different colored liquids. The center one is plain water. The right side there was a blue dye that has been added, and the left is blue dye and the right is red dye. You can see that the water has risen up, and you can see the leaves that have gotten that blue and red color on either side of normal water. Now this shows how plants actually take up water. Very tall plants, very tall trees, right from their roots to the tip of their leaf, the water flows by capillary action against gravity. Now this is a beautiful capture about capillary rise in different diameters of capillary tube. You can see here, as the radius of the capillary tube decreases, the ascent, the height to which the liquid rises also increases. Isn't that what we saw in our, formula for ascent? H is equal to, we saw it as 2t by R rho g cos theta. So the radius of the capillary tube is in the denominator. So that means it is inversely proportional to the height. Lesser the radius, greater is the height to which the liquid will climb up to. So you can see here, as and when they have reduced the height of the radius of the capillary tube, the height to which the liquid is climbing has kept on increasing. Another proof of our H is equal to 2t by R rho g cos theta. Now, this is another example. This is what you saw in the previous case where there were different colored liquids. Now, this is again the same thing where the liquid, where they have added a little bit of food color. It could either be milk or water. You can see that the liquid has risen up the tissue paper by capillary action. Now, this is another one where water rises up the sponge. There is this is your scrubber which you use in your kitchens. So the water moves up because of the fine capillary tubules that are present inside the sponge, and that again shows capillary rise in your sponges. Now this is now these pens are almost extinct in the student community these days, but these were extremely in use in large numbers when we were students. We call this as an ink pen, if you know. And you can see over here that there is a small cut at the tip of the nib. We call this as the nib of the ink pen. Right now, this extremely fine cut is given so that the ink flows from this. There is a small region where ink is stored over here. So the ink flows through the tip for smoother writing on your paper. So that's an ink pen where again capillary action actually plays a role in the writing of your pen. I can keep going on, but I would leave more examples to you. Water rising up your bath towel when you hold it, a particular only one tip of it in the bath tub, then water rises up the towel, and water rising up in various cases, not necessarily towels and paper towels and cloth towels. You can see various instances where water rises up by capillary action. Is that clear? Now, I'm going to end this lesson with a small question to you. Now, we saw that water rises up in a capillary tube to a particular height. In order to balance, this height will balance the pressure that has lowered because of the concave meniscus. Now, what if, listen to my question, what if you insert a capillary tube of insufficient height inside the liquid column? Let us assume that it has to climb to a height h, right? What if your capillary tube is of only height h dash, which is much lesser than h? What are you going to expect to happen when such a tube of insufficient height is inserted in a water column? Do you think water is going to flow up in the form of a fountain? That's going to be a question that you are going to ponder over. That's going to be the question you are going to research and find out what's going to happen if a capillary tube of insufficient height is going to be inserted in a liquid column. Hope you understood the examples and hope you will search for more examples about capillary action and get ready for your next activity to update your DP. Till then, stay safe.